Thank you. So I'm pleased to be able to present some of our work, and I thought I only had 5 minutes, but I think I have too many slides anyway, so that's okay. So I'm going to talk about microbiology. So I'm a microbiologist, but maybe a little bit strange for people who think about microbiology. Most of the applications in our lab are not medical focused. Although we have a couple, but most of the applications are not medically focused. I'm going to be talking about something called metagenomics, and metagenomics is basically looking at those organisms that you are unable to cultivate and using DNA-based methods, DNA sequence-based methods to access the information that those organisms have. And so a number of years ago, less than 10 years ago, there was this report that came out from the US National Research Council that is really worth taking a look at if you're interested in this hidden world of microbes, the organisms that you're unable to cultivate. And one of their final sentences was this. They proclaimed that the emerging field of metagenomics presents the greatest opportunity perhaps since the invention of the microscope to revolutionize understanding of the living world. So we all know that the microscope was an important invention that really allowed us to first see the microbes that nobody had any idea were existing. Well, even though we're able to see those microbes with the microscope, a lot of the information in those microbes is not available to us using traditional techniques. So the traditional techniques are, aside from just looking at them under the microscope, they are cultivating those organisms. And most of the organisms that you try and cultivate, they will not grow. So there's no way that you can study them using cultivation-based techniques. So what we do is we go directly and extract the DNA from microbial communities. Part of this is part of this Canadian Metamicrobiome Library, where we isolate DNA from soil samples that are collected from across Canada. And so we've got a variety of different soil samples that are quite diverse. We have Arctic tundra, we've got agricultural soil, northern peatlands, oil sands. I always catch myself there. Compost, swamp, and boreal coniferous forests. So those are some examples of the soils that where we sample. And as you can see, these are very, very different environments, and they have very different organisms, although they are all soils. And you can do a taxonomic profile where you look at a marker gene, the 16S gene that all bacteria have, and that will tell you which organisms are present. And so these are the major groups of bacteria, and you can see that the distribution is quite different. Soils are probably the most rich environments, the most diverse environments as far as the types of microbes that they carry. But even so, there is a different distribution. You can see that the oil sands is a little bit different than almost all of the other soils. Okay, and that really is consistent with the characteristics of the oil sands. So what do we do then? We isolate the DNA, so we're very careful to clean it up as much as we can because there's a lot of inhibitors in soil. Soil is very dirty. And if we want to be able to clone those fragments, we have to remove the inhibitors. So we have these nice clean DNA that we can shear, and we then take vector DNA, which we can then linearize and clone those fragments into. And this is a map of the vector that we use. And it has these important sites here called COS sites. These are sites that are important for packaging these fragments into bacteriophage heads and tails. And so you have these bacteriophage heads and tails. This is a commercial product. It's been used for many, many years in molecular biology. And basically what happens is it snips out this region here to here, from one COS site to the other. And this then regenerates our vector DNA, which is able to replicate in the cell. And it now has this random DNA, which is from an organism's genome that was in our original sample. It circularizes it, and it then inserts it into bacterial cells, and those bacterial cells can then form colonies. We then collect those, so each of these colonies has a vector that contains an insert DNA, and each insert DNA is from a different part of an organism's genome. Okay, so you've got thousands of different genomes represented in these colonies, and we've got thousands of these plates with thousands of colonies. We collect those clones and we save them as pools. So here's some examples of our libraries. So we've got a number of those soils that I showed you, and we also have human gut genome. This is basically just fecal samples that represent the human gut, a different type of soil, but important nonetheless. And you can see that we have fairly large fragments. So if each gene is about 1,000 base pairs, or 1 kilobase pair in size, you can see that each of these fragments has somewhere between about 25 to 40 kb average size. Okay? So 25 to 40 genes. And our largest one here has almost 9 million clones, so that's an equivalent of almost 80,000 genomes. So these are quite representative libraries. So I only have 2 minutes. I wanted to tell you an application of these libraries. So one of the things that we want to do is we want to be able to replace these fossil fuel-based plastics that don't degrade with plastics that can degrade. And we introduced the libraries into this strain of bacteria, which we've mutated so it can't make bioplastics. And you can see this one clone here is filled with bioplastics. And you can tell just visually with your eye because it's more opaque. And we find many of these colonies that are opaque like this. We sequence the DNA and we find that they have PHA synthase genes. These make a type of bioplastic called polyhydroxyalkanoate. Now the challenge is to make polyhydroxyalkanoates that have different compositions that will give them different physical and chemical properties. And so if you look at what the composition of the bioplastic, of the polymer, that is made with some of these clones, and this is just a selection, some examples, using different carbon sources. So here we have octanoate, here we have gluconic acid. So these clones are expressed in Pseudomonas, and you can see that they have, in these cases, they're all very short chain fatty acids that make up those monomers. Same thing here. But these ones here are interesting. They've got a mixture of monomers of different lengths. And these will give them different properties. So what our goal is to take our collection of metagenomic clones that contain different polymers and put them in different combinations and different genetic backgrounds in order to sort of mix and match and get different polymer composition that will give us different physical and chemical characteristics of polymers that can then potentially have commercial applications. So I'll stop there. I think my time is up. If you have any questions.
Really quick question for Trevor. So yeah, I'm amazed about this biopolymer research that you're doing. How expensive is it to make this polymer?
To make it? The big challenge is that you want to be able to make polymer that can replace a fossil fuel-based polymer, but for not twice or 10 times the cost, right? We're not there yet, but you have to remember that the fossil fuel-based polymers, that's a very mature technology. Eventually that price will go up and the price of these will go down. The other thing that you can do is you can target polymers or applications where the cost of the polymer is not as important as the final application.
Trevor, I believe they already have these biodegradable plastic ICI, but the problem is when they get into a landfill site, they don't get enough air to have the biodegradable, and you still have it there.
Yeah, so one interesting idea is a biological recycling. So you take these and you use them actually as feedstock to make other products.